structure of chromatin. We will also look into heterochromatin, euchromatin. We will look into histone and DNA acetylation, methylation, and histone deacetylation. It's a lovely morning today. I'm sitting outside and we're going to go through this module and we'll try to understand these concepts. So, looking at the chromatin structure, DNA chromatin structure refers to the complex of DNA and proteins found in the nucleus of eukaryotic cells. And now we'll go into a detailed overview. What are nucleosomes? They are the fundamental unit of chromatin. It consists of a segment of DNA which is wrapped around a core of histone proteins. Each nucleosome contains about 147 base pairs of DNA and a histone octamer, which includes 2 copies each of histones H2A, H2B, H3, and H4. The linker DNA is a stretch of DNA between nucleosomes, which varies in length but typically consists of about 20 to 60 base pairs. Histone H1, this histone protein, binds to the linker DNA, helping to compact the nucleosome array into a more condensed form. The 30 nanometer fiber nucleosomes further coil and stack to form a 30 nanometer fiber, which represents a more compact structure. This fiber can be further folded into higher order structures. There's also a loop domain. That's a 30 nanometer fiber loop that is there to anchor to a protein scaffold. If you look at the picture on the left, you can see the H1 histone protein. You can see the linker DNA. It almost looks like a clip that's holding the octamer together. And you can see the core DNA and you can see the octamers. There are those 4 little blue balls on the top and 4 blue balls underneath. And it consists of histones, namely H2A, H2B, H3, H4, and there's 2 of each. So you see 8 of them in total, which is nicely bundled up and made even more compact with the help of histone, which is an H1 protein. So this is very important, and I'd really like you to remember this one, that H1 is found outside of this core, the H1 histone. It is found outside of the core and it helps promote chromatin compaction. So there could be a question, and I think I'll be going through a question at the end of this module. This is a very important thing to know, that although you have the histones, the core, which is made up of H2A, H2B, H3, and H4, it's all compacted, and it's like this whole structure is kept in a compact way by the help of H1. So that green little button over there, that's H1, that's the H1 histone, and its job is to promote chromatin compaction. I've tried to make a diagram of a nucleus, and in there I've put in a bar body. There's a nucleolus, which is the very dark black circle. And then there's heterochromatin, which looks a bit darker than euchromatin. And I think euchromatin in this diagram is the lightest. So these colors are very important, because now we'll get into what is euchromatin and what is heterochromatin. We'll also look into the bar body. So euchromatin is a less condensed form. Euchromatin has a loosely packed structure. It allows for easy access to DNA. It appears as lightly stained regions under a microscope. The function is euchromatin contains genes that are actively being transcribed into RNA, making it the site of most gene expression. It's highly dynamic. The structure of the euchromatin can change, becoming more or less condensed depending on the cell's need for gene expression. The location. Euchromatin is usually found dispersed throughout the cell. Histone modifications. Euchromatin is often associated with acetylated histones, which correlate with active transcription. So when a histone is acetylated, it's exposing that part of the DNA, and that part of the DNA is called euchromatin, which is actively transcribing itself. Moving on, what is heterochromatin? Now, as you can see in the diagram, heterochromatin is slightly darker. It's a highly condensed structure. It has a very tightly packed structure. It makes DNA less accessible. It appears darkly stained under the microscope. The function is basically this area, heterochromatin. This is transcriptionally inactive. Heterochromatin contains genes that are not being actively transcribed. It plays a role in maintaining structural integrity and gene regulation. Heterochromatin is more stable and less dynamic compared to euchromatin. Where is heterochromatin found? It is found at the nuclear periphery and around the nucleolus. Heterochromatin is often located at the periphery of the nucleus and around the nucleolus. Histone modification. So, heterochromatin is methylated. Heterochromatin is frequently associated with methylated histones, which correlates with gene silencing. So, let's look at the summary of the differences between euchromatin and heterochromatin. When we look at the structure, it is loosely packed in euchromatin, and if you look at heterochromatin, it is tightly packed. The appearance is lightly stained for euchromatin and darkly stained for heterochromatin. Euchromatin is actively transcribed, whereas heterochromatin is transcriptionally inactive. Euchromatin is found throughout the nucleus, but heterochromatin is found near the periphery and around the nucleolus. Histone modifications. Histone acetylation makes that part of the DNA active, and that's euchromatin. And when the histones are methylated, it becomes a tightly compact structure. It becomes inactive, and that's heterochromatin. The gene expression is very high in euchromatin, and it's very low or none in heterochromatin. Euchromatin is a very dynamic structure, and heterochromatin is a very stable structure. Now let's look into the bar body, which you can see as mentioned in the diagram on the right side. A bar body, also known as a sex chromatin body, is an inactivated X chromosome found in the cells of female mammals. This inactivation is a normal process that occurs during early embryonic development to ensure that females, like males, have one functional X chromosome per cell. Here are some key points about bar bodies. So, X inactivation. In female mammals, one of the two X chromosomes is randomly inactivated in each cell. This process is known as lionization. It is named after the geneticist Mary Lyon, who first proposed it. The mechanism: the inactivated X chromosome becomes highly condensed and is transcriptionally inactive, forming a bar body that can only be seen under a microscope. The structure and location. Under a light microscope, a bar body appears as a dense, dark, staining spot at the edge of the nucleus. In the nucleus, the bar bodies are usually found attached to the nuclear envelope. The function and importance. Dosage compensation. The primary role of X inactivation is to equalize the expression of X-linked genes between males who are XY and females that are XX. Mosaicism, because X inactivation is random, females are mosaics for X-linked genes, meaning some cells express genes from one X chromosome while others express genes from the other. Now let's do a review of whatever we've done so far, and we'll look at an image which incorporates all of these terms that we've just come across. So I'll start with number 1. DNA exists in the condensed chromatin form to fit in the nucleus. We can see that in the nucleus. You can see that the DNA is there in a condensed form and it's fitting inside the nucleus. DNA loops twice around a histone octamer to form a nucleosome beads on a string. If you remember the diagram that I showed you in slide 1, you saw that you had the octamer in the middle and DNA looped twice around to form a bead on a string appearance. So over here, if you look at the diagram, you can see the histones. You can see the histones and you can see that they're looping twice. You can see that there's a twice of a loop made around each of those histone octamers. Okay, so H1 binds to the nucleosome and to linker DNA. So you know that H1 is binding to this nucleosome as well as to the linker DNA, thereby stabilizing the chromatin fiber. DNA has a negative charge from phosphate groups. And histones are large. They are large. Don't forget that. Even in the image I showed you, those blue balloons, those air blue balloons, they look quite large compared to um, the DNA. So DNA has negative charge from phosphate groups. Histones are large and have positive charge from lysine and arginine. So how does the the histones get its positive charge? It gets it from lysine and arginine. In mitosis, DNA condenses to form chromosomes. So here you can see on the image, that's a chromosome. DNA and histone synthesis occurs during S phase of the cell cycle. That's a very important thing to remember. There could be a question where they just ask you, when does DNA and histone synthesis take place? And it occurs during the S phase of the cell. Mitochondria, they have their own DNA, which is circular and does not utilize histones. I have a very nice presentation on how mitochondria undergo transcription and translation inside the matrix. Please have a look at that if you want to know more about mitochondria's own circular DNA and why they don't have histones. Okay, so let's just look at the image. So here you have the nucleus, and then you can see there's a detailed diagram of the chromosome. You can see the telomeres, the centromere, and you can see heterochromatin. It is highly methylated. It is very condensed, tightly packed. It's very stable. And here you have histone deacetylation. It's a supercoiled structure. And here you have euchromatin, which has undergone histone acetylation. It's very loosely packed structure, is very dynamic. And here on the side, I've mentioned the different histones that form the octamer. It's H2A, H2B, H3, H4, and it's 2 of each. And H1 is found outside of this core, and it helps promote chromatin compaction. Okay, moving on. So let's look into histone methylation. What happens in histone methylation? There's an addition of methyl groups. Methylation involves the addition of 1, 2, or 3 methyl groups to the lysine or arginine residues of histone proteins. The enzymes that are involved are histone methyl transferases. They add methyl groups, while histone demethylases, they remove them. The effects on gene expression. The effect of methylation on gene expression depends on the specific amino acid residues being methylated. For example, methylations of histone H3 at lysine is generally associated with active transcription, while methylation of histone H3 at lysine 9 and lysine 27 is associated with transcriptional regression. Histone methylation can either activate or repress gene expression depending on the location and the context of the methylation. Chromatin state can induce either euchromatin or heterochromatin depending on the specific methylation markers. Chromatin can either be in an open euchromatin or closed heterochromatin state. So histone acetylation, moving on to histone acetylation, it is a process of adding acetyl groups. Acetylation involves the addition of acetyl groups to the lysine residues of histone proteins. The enzymes involved are histone acetyl transferases and acetyl groups. The effects on gene expression, activation. Histone acetylation is generally associated with gene activation. It neutralizes the positive charge of lysine. It reduces the affinity between histones and DNA, resulting in a more relaxed chromatin structure that is accessible to transcription factors. Increased accessibility. The relaxed chromatin structure allows transcriptional machinery to access DNA more easily, promoting gene expression. Euchromatin. Acetylation is commonly associated with euchromatin, where DNA is more open and actively transcribed. So here I have a fun fact. The fun fact is that thyroid hormone synthesis is influenced by the acetylation of the thyroid hormone receptor, which can impact the expression of genes involved in the process. Histone acetylation, a key epigenetic modification, typically makes DNA more accessible and active for transcription. This means that when histone proteins around which DNA is wrapped are acetylated, they lose their positive charge, reducing their affinity for the negatively charged DNA. This loosens the chromatin structure, making the DNA more accessible to transcription factors and other machinery necessary for gene transcription. Consequently, acetylation of the thyroid hormone receptor or associated histones can lead to increased transcription of genes necessary for thyroid hormone synthesis and activity. Histone deacetylation. Here, the acetyl groups are removed from the histone proteins. The enzymes involved are histone deacetylases. They remove the acetyl groups. Histone deacetylation is generally associated with gene repression. It restores the positive charge on lysine, increasing the affinity between histones and DNA, leading to a more compact chromatin structure that is less accessible to transcriptional machinery. The compacted chromatin structure limits the access of transcription factors to DNA, thereby repressing gene expression. Heterochromatin deacetylation is typically associated with heterochromatin, where DNA is tightly packed and transcriptionally inactive. Here we have another fun fact. Histone deacetylation may play a role in altered gene expression associated with Huntington's disease. When histones are deacetylated, they regain a positive charge, which increases their affinity for the negatively charged DNA. This process tightens the chromatin structure, making the DNA less accessible to transcription factors and other machinery necessary for gene expression, effectively deactivating or silencing genes. Consequently, histone deacetylation can lead to reduced expression of genes that may be crucial for normal cellular function, contributing to the pathogenesis of Huntington's disease. Let's just look at Huntington's disease because it's easier to understand the concept and know the disease. What happens in Huntington's disease? In Huntington's disease, there's a loss of GABAergic neurons in the striatum. It's an autosomal dominant trinucleotide CAG repeat expansion in Huntington HTT gene on chromosome 4. There's a toxic gain of function. Symptoms typically manifest between age 30 and 50. You have chorea, athetosis, aggression, depression, dementia. Sometimes it is also initially mistaken for substance use. Atrophy of caudate and putamen with ex vacuo ventriculomegaly, dopamine, GABA, and acetylcholine in the brain. What happens here is that dopamine will increase, GABA and acetylcholine in the brain decreases in this condition. There's a neuronal death via NMDA receptor binding and glutamate excitotoxicity. Sorry about that. Anticipation results from expansion of CAG repeats. So there's a phenomena here that happens is that there's anticipation, and with each successive generation, the number of CAG repeats will increase and the onset of symptoms will be earlier than what the previous generation experienced. Caudate loses acetylcholine and GABA, and there's an increase in dopamine. Ex vacuo ventriculomegaly appearance of CSF on imaging, but is actually due to brain tissue and neuronal atrophy. Example, Alzheimer's disease, advanced HIV, frontotemporal dementia, Huntington's disease. The ICP is normal, but the NPH triad is not seen. Deutetrabenazine and tetrabenazine, they inhibit vesicular monoamine transporter VMAT. They decrease dopamine vesicle packaging and release, which may be used for Huntington's chorea and tardive dyskinesia. And here you can see the image on the side, which shows ex vacuo ventriculomegaly, which happens in Huntington's disease. Now this is quite important. Even if you've skipped the entire video where I've discussed histone methylation, acetylation, and deacetylation, even just knowing this will be quite helpful. So histone methylation is when methyl groups are added. The enzymes are methyl transferases and demethylases. It can activate or repress depending on the content of gene expression. In the chromatin state under histone methylation it can result in either euchromatin or heterochromatin. Histone acetylation, there's an addition of acetyl groups to the histones. The enzymes are acetyl transferases. The gene expression generally activates gene expression, and the chromatin state, it is always associated with euchromatin, open chromatin, which is transcriptionally ready to be transcribed. Histone deacetylation, the process is removal of acetyl groups. Enzymes are deacetylases. Gene expression is generally repression of gene expression. Chromatin state, it is associated with heterochromatin, closed chromatin. so it's transcriptionally inactive. Now let's look into DNA methylation. DNA methylation involves the addition of a methyl group to the 5 carbon of the cytosine ring in a CpG dinucleotide context. Cytosine followed by guanine. The enzymes involved are DNA methyltransferases. They're responsible for adding methyl groups to DNA. The effects of the gene transcription is that there's gene silencing. Methylation typically represses gene expression by preventing the binding of transcription factors and recruiting proteins that compact the chromatin, making it less accessible. The long-term regulation is that DNA methylation is a stable modification that can be maintained throughout cell division, playing a crucial role in long-term gene regulation, genomic imprinting, and X chromosome inactivation. The chromatin state is associated with heterochromatin. These high levels of DNA methylation are usually associated with the condensed chromatin state, which is transcriptionally inactive. When you think about DNA methylation, a syndrome should come to your mind, and that syndrome is Fragile X syndrome. The FMR1 gene produces a protein called FMRP, which is Fragile X mental retardation protein. FMRP, it plays a crucial role in the development and function of the nervous system. It is involved in the regulation of synaptic connections, and it is important for normal cognitive development and functioning. Mutations in the FMR1 gene, such as expansion of the CGG trinucleotide repeats, can lead to Fragile X syndrome, which is a genetic condition causing intellectual disability, behavioral and learning challenges, and various physical characteristics. Let's just look into the disease a little bit more so that you can remember it even better. So, Fragile X syndrome is transmitted through both parents. Children of affected mothers each have a 50% chance of being affected. 100% of daughters and 0% of sons of affected fathers will be affected because, of course, the father is transmitting the Y gene to the sons. X-linked dominant inheritance. The FMR1 gene produces a protein called FMRP, which is Fragile X mental retardation protein. FMRP plays a crucial role in the development and function of the nervous system. It is involved in the regulation of synaptic connections, and it is important for normal cognitive development and functioning. Methylation within the gene promoter, CpG islands, typically silences gene transcription. Dysregulated DNA methylation is implicated in Fragile X syndrome. Trinucleotide repeats in the FMR1 gene with hypermethylation of cytosine residues. Most common inherited cause of intellectual disability, Down syndrome, is most common genetic cause, but most cases occur sporadically. Trinucleotide repeat expansion CGG occurs during oogenesis. Remember, this is really important. The trinucleotide repeat expansion CGG, it occurs during oogenesis. Pre-mutation, where there's only 50 to 200 repeats, in that condition you'll have tremor, you'll have ataxia and first degree ovarian insufficiency. If it's a full mutation where there are greater than 200 repeats, you'll have post-pubertal macroorchidism, enlarged testes, long face with large jaw, large everted ears, autism, mitral valve prolapse, hypermobile joints, and self-mutilation is common. So in the full mutation, which are greater than 200 repeats of the CGG, what you'll see is enlarged testes. Very important. It's a very important clue in a question stem. Enlarged testes. You'll also see very long face, very large jaw, and these are the most important ones. The large testes, the long face, and the long jaw. This usually be mentioned in the stem. Now, what is DNA acetylation? Acetylation is the addition of acetyl groups to histones. DNA acetylation is an indirect term referring to histone acetylation, which involves the addition of acetyl groups to the lysine residues on histone proteins associated with DNA. The enzymes involved are histone acetyltransferases. They add acetyl groups to histones. The effects on gene expression. Gene activation. Acetylation neutralizes the positive charge on histones, reducing their interaction with the negatively charged DNA. This relaxed chromatin structure allows transcriptional machinery to access the DNA, thereby promoting gene expression. Short-term regulation, histone acetylation is a more dynamic and reversible modification, allowing for short-term regulation of gene expression in response to cellular signals. This is very, very important. Histone acetylation is it's dynamic, it's reversible modification, allowing for short-term regulation of gene expression in response to cellular signals. So the chromatin state, which is associated with DNA acetylation, is euchromatin. High levels of histone acetylation are typically associated with an open chromatin state, which is transcriptionally active. So let's look at the features of DNA methylation and DNA acetylation. Then we'll move on to some questions. I think I have about 2 in this presentation. The process is the addition of methyl groups to DNA, which is added to cytosine in DNA methylation, and there's an addition of acetyl groups to histones in DNA acetylation. The enzymes are DNA methyl transferases, and in DNA acetylation, the enzyme is histone acetyl transferases. The gene expression typically represses gene expression in methylation, and it typically activates gene expression in acetylation. The chromatin state, of course, DNA methylation is associated with heterochromatin. It's condensed, and while DNA methylation is associated with heterochromatin, euchromatin is associated with DNA acetylation. The key functions: long-term gene regulation, development, and genomic stability. And obviously, DNA acetylation would revolve around transcription and chromatin remodeling. So here we go. This is the question. If you've listened to the whole presentation, you will honestly be able to answer these questions without even thinking for a second. So histological analysis of a woman's epithelial cells are each found to contain a condensed body composed of heavily methylated DNA at the periphery of the nucleus. This region of DNA is most likely associated with which of the following genetic functions or findings? So what would be the answer? Is it histone acetylation or is it low transcriptional activity? If you guys are saying low transcriptional activity, you guys are absolutely correct, because in genetically normal females, 46XS, one X chromosome is normally randomly deactivated in each cell during early embryonic development. This X inactivation is called lionization. It is maintained across cell division, resulting in clusters of cells throughout the body that express either the maternal or paternal X chromosome. This mosaic pattern of X chromosome expression generally prevents X-linked recessive conditions from manifesting in female carriers. And we'll go into a little bit more details about this. So the fun fact is that a small proportion of genes remain transcriptionally active on the inactivated X chromosome. For this reason, inheritance of an abnormal number of X chromosomes can cause clinical manifestations due to a gene dosage effect, as seen with Turner syndrome, which is 45XO. There's only one X chromosome here. And Klinefelter's, which is 47XXY, which has two X's and one Y. In rare cases, skewed lionization, uneven inactivation of maternal or paternal X chromosomes due to chance alone, may result in females developing an X-linked recessive condition, example classic hemophilia. So in genetically normal females, one X chromosome is normally randomly deactivated in each cell during early embryonic development. X inactivation, lionization is maintained across cell division, resulting in clusters of cells throughout the body that express either the maternal or the paternal X chromosome. This mosaic pattern of X chromosome expression generally prevents X-linked recessive conditions from manifesting in female carriers. The process of lionization converts the inactive X chromosome into a condensed heterochromatin, which can be identified on microscopy as a compact body at the periphery of the nucleus, also called bar body. Heterochromatin consists of heavily methylated DNA, example cytosine converted to methyl cytosine, and deacetylated histones, which causes it to have a low level of transcriptional activity. There you go, that was your answer, why the answer was a low level of transcriptional activity. Moving on to question number 2. During interphase of the cell cycle, electron microscopy of a eukaryotic cell reveals 10 nanometer thick chromatin fibers with a bead on a string structure. These fibers are then extracted and treated with an endonuclease, which selectively cleaves the string section of the chromatin. Further analysis of the beads shows they consist of DNA wrapped around a protein core. Which of the following proteins is most likely located outside this core and assists in chromatin compaction? Now, the clue here is that it's literally giving you the answer. It says which of the following proteins is most likely located outside this core. We spoke about the octamer, right? And we spoke about the core. It has 8 of these histone proteins. But there's one of these histone proteins that's outside. And it assists in chromatin compaction. If you're thinking H1, then you're correct. That is the answer. H1 is found outside of this core and helps promote chromatin compaction. You can see the image on the right lower side of the screen. The octamer of core histones, it consists of H2A, H2B, H3, H4. There are 4 of them on top and 4 of them on the below. Histone 1 is like a clip that is closing this octamer, like a little bow on a present, and it is found outside of this core and it helps promote chromatin compaction. The fact that this histone H1 is green in color, that should also help you form a memory of this, that it's outside, all the blue ones are inside and the green one is outside. So in this question, I think the only thing that was a little confusing was SNRNPs. If you didn't know what that was, SNRNPs are small nuclear ribonucleoproteins. They help to splice out introns from pre-mRNA, forming mature messenger RNA. And that is the end of this presentation.